the summer of 2006, Rahman Bayrami receives a diagnosis that changes everything: prostate cancer. After initial success, the cancer seems to be defeated, but 5 years later, it returns. What follows is a long journey marked by hope, setbacks, and new therapies. The cancer spreads to other parts of his body, but Rahman Bahrami remains steadfast. His positive outlook gives him strength.
Es gibt einfach Leben mit Krebs, aber mit der Zeit, wenn du denkst und respektierst die Therapie, dann du hast keine mehr Krankheit, hast einfach positiv denken. Ich sage das in Englisch: If you can change your mind, you can change your life. Never give up. Keep going. You're gonna win.
When standard therapies fail, prostate cancer becomes a particular challenge. At the University Hospital Basel, Professor Damian Wild and his team are actively searching for new approaches. In collaboration with the ETH-Domäne, they are now testing a newly developed radiopharmaceutical in a clinical trial. An interdisciplinary project with a clear goal: tailored therapies for patients with metastatic prostate cancer that no longer respond to conventional treatments.
Etwa 1400 Patienten versterben am Prostakarzinom in der Schweiz jährlich. Aus diesem Grund ist es notwendig und wichtig, bessere Therapien zu entwickeln. Die neu entwickelte Therapie hat das Potenzial, die Ausbreitung, die Metastasierung beim Prostakarzinom besser zu kontrollieren als aktuell verfügbare Therapien. Nur ein geringer Anteil der neu entwickelten Therapien ist effizient genug, um klinisch evaluiert zu werden.
The new radiopharmaceutical, developed here at the Paul Scherrer Institute within the ETH-Domäne, has successfully passed all key milestones to enter clinical testing. It is based on a newly developed radioligand and an improved radioisotope, a combination that enables not only precise imaging of disease-related structures in the body, but also targeted therapy. At the Center for Radiopharmaceutical Sciences, Professor Christina Müller played a leading role in its development.
Diese klinische Translation unserer neuen Entwicklung zu ermöglichen, haben wir 10 Jahre lang in meiner Forschungsgruppe gearbeitet. Und zwar haben wir ein Ziel verfolgt, bei dem wir neue Radioliganden entwickeln wollten, die eine verbesserte Tumoranreicherung zeigten. Dies haben wir erreicht, indem wir einerseits neue Liganden hergestellt haben und andererseits ein neues Nuklid eingesetzt haben, das hier am Paul Scherrer Institut durch meine Kollegen in der Radiochemie erstmals produziert wurde. Diese Kombination von einem neuen Radioliganden und einem neuen Nuklid wurde dann präklinisch im Detail untersucht im Rahmen eines PHRT-Projektes und wir konnten wirklich zeigen, dass die Therapie in Mausversuch sehr gut wirkt.
Professor Müller knows bench-to-bedside research is teamwork, across disciplines, united by a common goal: safety and efficacy.
An diesem Projekt war für mich speziell interessant und bereichernd, dass eben sehr viele Leute involviert waren. Ich hatte eine sehr intensive Zusammenarbeit mit meinem eigenen Team, aber auch mit anderen Forschungsgruppen hier am Paul Scherrer Institut und natürlich auch über das Institut hinaus und jetzt auch immer mehr mit den Klinikern, die dieses Produkt in den Patienten anwenden.
The Paul Scherrer Institute provides a unique environment for the development of innovative radiopharmaceuticals. Professor Roger Schiebli, head of the Center for Radiopharmaceutical Sciences, explains what makes PSI so special and how it combines research and production at the highest level.
Radiopharmazeutische Forschung hat am PSI begonnen. Dank diesen großen Anlagen, die wir hier haben, diese speziellen Anlagen, Beschleunigeranlagen, Neutronenquellen, können wir neue Isotope, Radioisotope für die Medizinalanwendung herstellen. Und wir haben gleichzeitig die Möglichkeit, diese Substanzen auch noch für den Patienten herzustellen. Also wir können vom Labortisch bis zum Patientenbett können wir die Substanzen hier entwickeln und auch zur Verfügung stellen.
Thanks to PSI's comprehensive expertise, the production and separation of new radioisotopes take place in so-called hot cells, shielded areas where high levels of radioactivity can be handled safely. Once purified and quality checked, the isotopes are incorporated into the drug's carrier molecules, which are produced in sterile cleanroom environments with high-quality standards. In the next step, they are ready for use in patients. The new medication is currently in an early phase of clinical testing. Dr. Aline Kiringdel from the University Hospital Basel is overseeing the trial and explains the stages this new medication passes through in clinical research.
Our study is a phase 1 clinical trial designed to translate a new radiopharmaceutical from preclinical development to human medicine. It will be conducted in 2 parts. Initially, in phase 1A, we'll do a head-to-head comparison between this new radiopharmaceutical and the standard at very low activity, so we can identify any unexpected side effects, if there is. Following this, we'll conduct a dose escalation study so we can identify and to understand better the performance of these new radiopharmaceuticals. The final goal of the study is to see which is this optimal biologic activity that can be checked in patients safely for a maximum efficacy of the treatment.
To gain deeper insights, additional blood analyses are being conducted in the laboratory of Professor Nicola Accetto at ETH Zurich. The goal is to better understand the course of treatment and to obtain indications of how individual patients respond to the new therapy.
We received samples from the University Hospital Basel here at ETH Zurich. We process them with a mixture of microfluidic and robotics technologies to isolate circulating tumor cells. And these circulating tumor cells in blood samples, first of all, they are profiled for their morphology, and we understand whether they travel as single cells or as clusters of cells, which is very important. And then with robotic tools, we isolate them 1 cell at a time, and we do gene expression analysis. So we try and gain insights into genes that are upregulated or downregulated during treatment. For example, a key gene we are interested in is PSMA, prostate-specific membrane antigen. This is the target of the therapy that these patients receive, and it's very important for us to monitor the expression of this target during treatment to understand how patients respond to the drug.
Based on the results of the current study, larger patient groups are expected to be examined in the future. The journey for this kind of research is still ongoing, but has now taken an important step forward. For patients like Rahman Bayami, this new radiopharmaceutical offers hope for more targeted therapies and improved quality of life, and perhaps, with time, new perspectives in the fight against advanced prostate cancer.